welcome to the video on how to genetically engineer bacteria to produce human proteins using plasmids. So if we take a look, this is a picture of a bacterium, and a bacterium has a circular chromosome here, and they also carry with them supplemental circles of DNA called plasmids. What we're going to do to get bacteria to produce proteins, human proteins, that we want it to produce is we're going to insert human genes into these plasmids and then force the bacteria to take the plasmid up from the environment, and then the bacteria will express that plasmid by transcribing and translating the DNA in the plasmid. Now the question is, why does this work? Why can we take this red circle, which represents a circular plasmid, and why can we take that plasmid, cut it open with the restriction enzyme, and then cut the gene of interest, which this green line represents, with the same restriction enzyme, and if we put them together, the gene of interest cut with the restriction enzyme will anneal with the plasmid that's been cut open with the same restriction enzyme. And then we can take this plasmid and insert it into bacteria, and the bacteria is able to take this green gene here and transcribe and translate it into protein. How is it even possible? At a very elementary level, this is possible because the process of making proteins, the process of protein synthesis, of transcribing and translating DNA, and the DNA code is an evolutionarily conserved process. Every organism, pretty much, the DNA codes for proteins, and the proteins are transcribed and translated in a similar process. It's slightly different in prokaryotes versus eukaryotes, but the result is the same. You take a DNA molecule, turn it into RNA, and then turn that RNA to protein at the ribosome. Not only that, this genetic code, which we spent with these codons on the mRNA, is universal. Okay? This code is read the same way in us as it is in bananas, as it is in bacteria, as it is in monkeys, as it is in Kaiser. Okay? He's right here, so that's why I said his name. So, if you insert an mRNA that has the codon UUU in every single organism, it's going to call for the amino acid phenylalanine, or Phe. The code is universal and read the same way in all organisms. It's one of those evolutionarily conserved processes that the AP exam loves. We're now going to go step by step in the process of making a plasmid and inserting it into bacteria, and then growing that plasmid and getting the bacteria that you want, getting the protein out of the bacteria that it just produced. Okay, we're going to go step by step. The steps are going to appear, and we're actually going to go to further slides, because there's further slides that sort of elaborate on the steps. Okay, so the first step, okay, is to open the plasmid by cutting it with a restriction enzyme. Next week sometime, depending upon all this snow ridiculousness, we are going to actually simulate this. So this will make a little bit more sense after we simulate this. But the first step is to open the plasmid, remember it's a circle, and cut it with a restriction enzyme once so that it opens up. Okay? The next step is to isolate the gene of interest, which we're not going to talk about how you do that, and cut it on either side of the gene using the same restriction enzyme that you cut the plasmid with. So the restriction enzyme here and the restriction enzyme here, they need to be the same. Okay? Now the question is why? Why does the restriction enzyme here and the restriction enzyme cut by the plasmid and the restriction enzyme cut by the gene of interest, why do they need to be the same? This image, which is on the next slide, helps to explain this a little bit. So this here, don't look at the plasmid for a second. Don't look down here. Pretend this is not here. Okay? This is the target gene. So this blue spot right here, this contains the sequence of the gene that you want to put inside the plasmid to get it into the bacteria. The first thing you're gonna cut this target gene with a restriction enzyme. And you notice here, okay, it leaves the sticky end AATT, and here it leaves the sticky end TTAA. Okay? If you want to get this gene into the plasmid, you have to cut the plasmid once with the same restriction enzyme. And you'll see that it has the sticky ends TTAA and AATT over here. Now, this is getting to step 3, which we'll go back to the text in a second. Okay? But if you now place this piece of DNA with this piece of DNA, you'll see that this sticky end, right here, can go right in here and is complementary to this sticky end. AATT can go here, like this sticky end will go here. I don't want to finish drawing it. You get the point, right? Those are complementary. Same thing here. This sticky end right here is complementary to this sticky end. And this target gene will then be able to be inserted in this spot right here, because the sticky ends create complementary base pairs that can hydrogen bond together. Okay, we're now going to go back to the steps. So we learned here, it says isolate the gene of interest and cut it on either side using the same restriction enzyme. The reason why you need to use the same restriction enzymes is that if you don't, then the sticky ends won't be complementary. And then you won't be able to, the sticky ends won't be complementary and you won't be able to put them together. So the point of using the same restriction enzyme why you have to is it makes the sticky ends complementary. Okay? Once you've done this, okay, you're going to need to place the cut plasmid and the gene of interest in a tube with DNA ligase. And what's going to happen is that the sticky ends of the plasmid and the sticky ends of the gene are going to anneal, because they're complementary and can hydrogen bond, and then DNA ligase is going to bond the sugar phosphate backbones together of them, so you have a intact DNA strand. Okay? Again, what should happen, like was what I just said, is that because the sticky ends are complementary, the plasmid and gene of interest are going to hydrogen bond to each other, and then DNA ligase will glue together the phosphate sugar backbone. Once we have this, hopefully, when we're doing this, we're not just doing like one plasmid and one gene of interest. We're doing like thousands of plasmids and thousands of copies of the gene of interest. Hopefully, at this point, we have some plasmid with the gene of interest in it. Okay? We are then going to take the bacteria and place it in a tube with this new, what we call recombinant plasmid. Okay? It's recombinant because it now has the DNA of the bacteria and the DNA of the human in this case. Okay? So it's a recombinant plasmid because two different DNA sources have been combined. And we're going to do something known as heat shocking it. Okay? And heat shocking forces the bacteria to take up the plasmid from the environment. We're now going to take a look at this process a little bit. Alright, so this is the heat shocking process. Okay? So you have your cells in a tube. Okay? Don't worry about this ligated mixture. And you put some calcium chloride in it. So CaCl2. CaCl2. Okay? That makes the membrane, it loosens up the membrane a little bit. Okay, and you're going to place these cells that have the plasmid in the environment. So you have, in this tube, you have the bacteria, and then you have the plasmid that you just made floating around in this tube. Okay, and you're going to put some calcium chloride in it. And you're going to let them sit on ice for a bit. Minimum 5 minutes. Okay, then you're going to take this tube and walk it over to a hot water bath that's at about 42 degrees Celsius. And you're going to let them sit in it for 30 seconds. After the 30 seconds is up, you're going to immediately transfer it back into the ice. Okay? Now, do you need to remember this process? No. You're physically going to do this process at some point, depending upon the snow. Okay? But the point is this process of putting in calcium chloride on ice, then putting it at 42 degrees Celsius in a water bath, and then putting it back on ice, makes the membrane more fluid. It increases the permeability of the membrane, so therefore the bacteria is more likely to take up the plasmid. Okay. Now, at this point, okay, we hope, okay, that we have some bacteria with the plasmid in it. Okay. Our next step, number 5, and you can see all the steps now, okay, is to grow the bacteria on growth media on a petri dish. Growth media contains all the nutrients that bacteria need to live. Okay? And just to show you what this looks like, this is an example of a petri dish. Okay? Each one of these, if you take a look at this, is a plate. Those that like orange stuff or yellow stuff, I don't know, whatever you want to call it, is the bacteria. Each one of these dots is called a colony. And a colony of bacteria has about 1 million bacteria cells in it. Okay? Remember, this is a petri dish, and on the petri dish is some, what they call agar, that bacteria grow on, and it has all the nutrients they need to live. This petri dish, the bacteria overgrew so much you can't even see the individual colonies. When that happens, that's called a lawn of bacteria, like you mow your lawn, a lawn of bacteria. Okay? So after you have your bacteria, and you've heat shocked it, hopefully they have the plasmid in it, and then you're going to grow them on a petri dish like this. And we're going to do this all in the lab next week. Okay? Step 6, 7, and 8. Okay, 6 we're going to talk about in more detail next week. It's too complicated for this video. Once you've grown the bacteria, you need to select and screen for the bacteria that got the plasmid with the gene on it. You're going to see, okay, that there's going to be some bacteria that didn't get the plasmid, okay? We need to learn how to select only for those bacteria that got the plasmid, okay? The bacteria that got the plasmid will then produce the protein from the gene of interest, and their own proteins, just through the general process of transcription and translation. And then the last step after that is to purify the protein from the bacteria using something called chromatography, which we're not going to learn about. Protein chromatography, which is not on the AP curriculum. Okay? We are going to talk much more about step 6 next week. Okay? Like a whole day on step 6. And then we're going to do the entire process, okay, except for step 8 in the lab. Probably next week as well, if the weather holds out. Alright. Answer the guided questions and have a good night.